The last part of molecular basis of inheritance is DNA fingerprinting. It's also called as DNA typing or DNA profiling. This method was developed by Alan Jeffreys, a British, in 1985. For that, he was awarded a prize from the Royal Society's Copley Medal. So he was a recipient of Royal Society's Copley Medal in 2014. Now you know that all individuals have the DNA. The chemical nature of the DNA is almost the same, but there are millions of differences in the base pair sequences, or in the sequences, the chemical base pairs, in the sequence of base pairs. Each of us is different in sequences except the twins, particularly the identical twins. So each of us, except you see that one, the identical twins, we are differing from each other genetically. We have nearly about 23 pairs of chromosomes and nearly about 1.5 million pairs of genes. 1.5 million pairs of genes. As per actually the calculation is given, 1.5 million pairs of genes in these 23 pairs of chromosomes. Million pairs of genes. This is one. You know the meaning of a gene. A gene is nothing but a segment of DNA that differ in nucleotide sequences between one individual to another. Not only the gene or DNA is coding for, you know that what the proteins, but not all the segments of DNA are coding for proteins. Some of the DNA segments actually acting as regulating the gene expression. So we have some segments of DNA, number one. So not all the segments of DNA are coding for proteins. Some DNA segments are acting as regulators. Some DNA segments are acting as the intervening sequences. These intervening sequences what is studied already in the case of split gene, introns. And some, just actually the segments of DNA acts as repetitive sequences. So, the meaning for that one, not the entire DNA is coding for a protein. Some segments of DNA are acting as the regulators regulating the process. Some are acting as intervening sequences, what are called the introns, non-coding sequences. Some segments are in the form of repetitive sequences. Now these repetitive sequences are nothing but the repetitive DNA. For DNA fingerprinting, the repetitive DNA sequence is specific for a person. The repetitive DNA sequence is specific for a person. These repetitive DNA sequences are together called VNTR. These repetitive sequences of DNA are called VNTR. Normally we can say sample DNA. What's the meaning of this VNTR? So, in DNAs or in what is called actually that is DNA fingerprinting. Now the repetitive sequences are specific for a person. Now these repetitive sequences are called VNTR. The expansion for that one, variable number of tandem repeats. Variable number of tandem repeats. We have certain stretch of DNA is being repeated many times. Such DNA segments are called repetitive sequences. So, and these sequences are called variable number of tandem repeats. Tandem means one after another. So the repetitive sequences are found either continuously one after another, then it's called tandem repeats, tandem, in a queue, one after another. Or in some cases, these repeats are found in a dispersed manner, not continuously. If the repeats are found continuously one after another, then it's called tandem repeats. These tandem repeats are VNTR for two persons show variations. So the VNTR is not same for two different persons. They are different from one another. And also they are being used as what is called the genetic markers. So the variable number of tandem repeats is not actually similar between two persons. In two persons, usually they show variations. That's why it's called variable number of tandem repeats. Okay. Now they act as genetic markers. Now I am taking what is called two person. This is person A. This is person B's DNA. I am taking some segments. Now, in the case of person A, now in one region we have the GT. There are 24 times. This is a sequence. GT is a sequence. It is being repeated 24 times in this person A in one chromosome, maybe 1 or 2, etc. Any chromosome active. Now another sequence ATGG, it is being repeated 16 times in the same chromosome in the DNA. Or in the same actually individual, the DNA shows what's called 66 repeats for ATT. So this is one individual. That individual has 24 repeats GT, 16 repeats ATT, repeated 16 times, and ATT 66 times. That is called 66 repeats. Now I am taking another individual, B, with the chromosome the same. Suppose we are taking the chromosome number 2 here also I am taking chromosome number 2. Comparing the same repeats. Now the same position. In this B individual, this GT is represented by 49 times. 49 repeats. Now this ATGG, the same number of repeats, no change. Then what about the third sequence, ATT? It is being repeated 51 times. And this is what's called variable number. So the tandem repeats are different from one individual to another. That's why it's called variable number of tandem repeats. The same repeat may not be present, that is between two individuals. Maybe present, you see that one in this one. So the ATGG, the repeats 16 times here also 16 times of another individual is possible, but they may not be similar with reference to these what is called the repeats. They are different. That's why I say we have millions of differences between the DNA sequences of one individual to another, or we can say the VNTR normally in two persons show variability or variations. And they are all actually genetic markers. This is what is called by represented what do you mean by variable number of tandem repeats. Now we have to go for the mechanism of, that is what is called DNA fingerprinting. DNA fingerprinting involves the identification of specific regions of DNA sequence. And such sequences are called repetitive DNA sequence. These sequences are called repetitive DNA sequence because a small stretch of DNA is being repeated many times, what I told you already, the VNTR. So a small stretch of DNA being repeated many times, hence it is called what is known as repetitive DNA sequence. Now such a repetitive DNA sequence are separated from what is called the bulk genomic DNA as what is called different peaks during centrifugation. By density gradient centrifugation, they are separated from the bulk genomic DNA, that is the remaining DNA, that is as different peaks, that is during the centrifugation by density gradient centrifugation. So the bulk genomic DNA forms major peaks like this. If you analyze that one, so these are all the major peaks, these are all the minor peaks. This is major peak. And this is a minor peak. So the bulk genomic DNA forms a major peak. And what we have, this repetitive DNA just normally forms actually smaller peaks, or we can say minor peaks. And such smaller peaks are called satellite DNA. So the another name for repetitive DNA is called satellite DNA. Now, what are the types of satellite DNA? And the classification or the criteria is based on three factors. On what basis the satellite DNA is classified? Number 1, the length of the segment. The criteria used for classification of satellite DNA. Number 1, length of segment. Length of, that is what is called the segment. Then base pair composition. Base pair composition. You know, you see that one normally we have A is forming a pair with the T, G is forming a pair with the C. So based on the richness of A and T and the richness of G and C. So base pair composition. Number 3, number of units. Number of units. So these are the 3 criteria used for the classification of satellite DNA. 1, the length of this repetitive DNA segment. Number 2, the base pair composition, whether it is rich in AT or rich in GC. And also the number of repeating units. I mentioned the units, nothing but the number of repeating units. And based on these criteria, we can classify the satellite DNA into either microsatellite or mini satellite. Micro or mini satellite. So this is what is called the criteria for classification of what we call the satellite DNA into either micro or mini, what is called satellite. Now we have to go for the properties of satellite DNA. What are the properties of satellite DNA? What are the properties of satellite DNA? So these satellites do not code for any proteins. That is one of the properties. But they form the largest proportion of human genome when compared to the normal component of genome. They represent the largest proportion. So they do not have any meaning for protein. But they form the largest proportion of human genome. They exhibit polymorphism. What is polymorphism? Variations at the genetic level due to mutation. It's called polymorphism. What is polymorphism? Variations at the genetic level due to mutation. So this is called what is known as polymorphism. Variations at the genetic level. The main cause of such variations, you see that one, that is mutation. So they exhibit polymorphism, different types. Then they form the basis for DNA fingerprinting. Without satellite DNA, we cannot make this DNA fingerprinting, which is unique for any human being. The DNA as well as what we call the DNA fingerprinting is unique for every human being. So these are the 4 important properties of DNA fingerprinting. What I can say, not DNA fingerprinting, the properties of satellite DNA. Do not code for protein, largest proportion of genome, exhibit polymorphism, and also they form the basis for DNA fingerprinting. In the case of forensic analysis, when there is a murder, then we want to know the suspect, whether he is a guilt or innocence. That can be analyzed. We can find out the nature of the persons who are involved in what we call the criminal activities, who is a guilt or innocence. And for that we are doing the DNA fingerprinting. What we are doing actually, the cells from the blood or the hair cells or from the skin cells, or is there any other genetic evidence from the crime scene? Crime scene means the place where the murder took place, where the incident took place. And that one is compared with what we have, that is the DNA of what we have the suspected criminal through VNTR pattern. Through VNTR pattern. So through VNTR pattern we are comparing the DNA collected from the crime scene with what we have the criminal suspect, whether he is a guilt or innocence. This is a major area in the case of forensic analysis. Now here I given actually 3 individuals. So see, the DNA is collected from the incident took place. That is a crime scene where the murder took place. And 2 persons are suspected. Individual A, individual B. Now here they are taking what we call the random chromosomes. Chromosome 2, they are taking chromosome 7 and chromosome what's called 16. In all the 3 cases, either from the DNA of crime scene or from suspect 1 or from suspect 2. They are taking, that is what we have, the chromosome 2, chromosome 7 and chromosome 16. Now they analyze what we have the tandem repeats. So through what is called the behavior pattern only we can compare the DNA of what is called the criminal suspect, okay, with that is the normal person or the DNA collected from the crime scene. Now if we are taking chromosome just 2, so normally we have a pair of chromosomes. One chromosome is paternal, another one is called maternal. One is coming from father, another one is coming from mother. These 2 chromosomes also differ in the number of tandem repeats because the 2 chromosomes are coming from 2 different parents. But normally the DNA of a father and the DNA of a son or a daughter with reference to tandem repeats same, or the DNA of mother with the son or daughter would be the same. But if you are taking the DNA tandem repeats, that is in the case of what is called father and mother chromosomes, they are differing because they are coming from 2 different parents. Now I am taking chromosome number 2. The number of tandem repeats here in paternal chromosome 3 and maternal chromosome 4, I represent 3, 4, the tandem repeats. And here in the case of suspect 2, the number of tandem repeats 11 and then 9. In the case of DNA from crime scene also 11 and 9. I am taking chromosome number 7. The number of tandem repeats 8 in the paternal chromosome, 7 in the maternal chromosome. And here also 8 and 7. Here also we have 8 and 7. So common for all the 3 individuals. One from the crime scene, another one from suspect 2 and another one from suspect 1. Now we are taking chromosome number 16. So in the case of individually suspect 1, they have 5 tandem repeats in paternal chromosome and 11 in the maternal chromosome. If we are taking what we have chromosome 16 in the case of crime scene and then in the case of that is suspect 2, in both the cases we have 5 and 4. So leaving the person what is called individual A, if you are comparing what we has the number of tandem repeats, it will be the same in chromosome number 2, in chromosome number 7 and chromosome number 16. Here 11, 9, here also 11 and 9. Here 8 and 7, here also 8 and 7. Here we have 5 and 4, here also 5 and 4. So the number of tandem repeats is normally matchable between individual B and the DNA from the crime scene. So here the culprit, that is a guilt, is nothing but individual B and not individual A, because the tandem repeats in chromosome number 7, though it is coinciding with the chromosome 7 of DNA from crime scene, in other 2 chromosomes there is no match. That's why we can say the person who just did the murder or the person who is guilt is nothing but individual B, the suspect 2 and not A. So it is being normally done through what is called actually DNA fingerprinting in the form of banding pattern. I will show the banding pattern also in a simple way in the next diagram. Here I drew the diagram of what is called the DNA from the crime scene. The number of repeats I mentioned about, you know that one 11, 9, 8, 7 and also 5, just 4. 5, 4, not 5, 5. That is 5, 4. Now when the DNA fingerprint has been made by means of radio-labeled technique, the bands obtained in the membrane or in the X-ray film show the similarities between C and D. Now this is a DNA from the crime scene. This is a suspect 2, that is S2, suspect 2, S1. If you are comparing the banding patterns, you see here the banding pattern in C and B same. Here also same. And if you are taking this one also same in all the three cases, but we are considering only more for B and C. Now the next one, you see that one same between B and C. Here the banding pattern is also same between B and C. So we concluded that normally the suspected person is nothing but actually B, whose banding pattern coincides with the banding pattern of what is called the DNA obtained from the crime scene. Hence he is considered as a guilt. It's not ignorance, it's a guilt for that one. Okay, this is about one of the uses what I mentioned about. So we are comparing the DNA of different persons. So if there is matching between what is called the crime scene DNA and the suspect DNA, then we can say that person is what is called a culprit. And also the same VNTR, that is actually banding pattern, the same VNTR banding pattern is also used to establish identity in the case of homicide victim. In the case of homicide victim, by using what is called either DNA or from the body itself. So we can also find out that is the identity of what is called the homicide victim, either from the DNA or from the body of that person itself. So that's about what we have actually the DNA fingerprinting, what we have the introductory part. The next one we have to go for what is called actually the various steps involved in making DNA fingerprinting. Okay, we'll discuss later.